Hi and welcome back to our GCP-Mindset channel and all topics on clinical research. In Part 8, we'll give some more information on statistical tests in clinical trials. More after the intro. Here you see a biometrician, as they probably prefer to regard themselves. According to the study design, the biometrician decides to use a t-test for statistical evaluation by means of a statistical procedure, represented here as the black box. The sample size is calculated from known parameters. In our example, an alpha error of 5% is acceptable, which means false positive results occur with a probability of only 5%. The definition of the beta error of 20% means the probability of discovering a real effect is 80%. The difference of interest is 5 units with a standard deviation of 7. Consequently, the black box calculates a sample size of n equals 64. The standard procedure for the sample size calculation is presented here. Typically, it is started from the question which sample size n is necessary to facilitate the verification of a clinically interesting d with a power of 1 minus beta. In this case, the d of clinical interest has to be determined first. Furthermore, the standard deviation of the relevant factor has to be identified based on literature references or a pilot study. Then the acceptable error limits have to be defined. The type 1 error is thereby typically set at 5%. In addition, you can see here the exceeding probability described as p-value. In statistical tests, the null hypothesis is rejected when the p-value is less than or equal to the significance level alpha. The beta error is usually set at 10 or 20%, corresponding to a test power of 80 or 90%, respectively. Consequently, an effect is discovered with a probability of 80 or 90%. The required sample size n is finally calculated with the help of formulas, software, tables, or graphical approximation. An example of sample size calculation. The issue here is finding the sample size necessary for the verification of a unilateral mean difference of 10 units, d, with a power of 80%. Alpha is 5% and beta is 20%. Thus, the sample size is exclusively dependent on the scattering. For less scattering values, 4 subjects are sufficient in order to achieve a significant result with the defined power. However, if the values scatter strongly, as many as 51 subjects are needed. Another example of sample size calculation. How many subjects are necessary for the verification of a unilateral mean difference of 10 units, d, with a standard deviation of 10 units, s. Because alpha has been set at 5% and the standard deviation is known, the sample size is dependent on the desired test power. A power of 80% only requires 14 subjects, a power of 99% even 33 subjects. Here you see a diagram showing the distribution of the measured values. The therapy group is shown in red and the control group is shown in blue. The mean value of the therapy group is at 135 and the mean value of the control group is at 155. The values of both groups do considerably overlap, which is typical for clinical studies. In our example, we have a sample size of 10 subjects in both groups with a standard deviation of 20 units. Now you can see the distributions with a sample size of 20 subjects in each group. The distribution of mean values is significantly narrower than the distribution of data. The distance of mean values is indicated by the t-value, which is calculated as a quotient of mean difference and standard deviation, which is subsequently multiplied by the square root of the total number of subjects. Thus, the sample size n plays a decisive role. So much for today on statistical tests in clinical trials. We hope that we could give you some interesting information and look forward to see you next time.